Let's take a departure from the normal, pristine, planted aquariums that we're all used to, and go see what's going on outside. In the strangest of places, if you live anywhere in the suburbs, a drainage ditch might be the only thing that's close to a natural waterway that you have. We're going to go deeper in these waters. We're going to test them with our lovely test strips. We're going to look at things underneath a microscope. And we're going to just sit here and observe all the biodiversity present in our drainage ditch. Because each one of these things can help us determine the health of our aquariums as well. Before the species tour, a word about what we are actually reading when we look at this ditch. Because in stream ecology, organisms are not merely inhabitants, they are data points. A species' presence is a vote for what that water can support. Its absence votes the other way. Crayfish require dissolved oxygen, a functioning food base, and water quality sufficient to sustain a crustacean with metabolic demands. You do not find crayfish in biologically dead water. Their presence is a certificate of minimum habitability, not excellence, but function. The damselfly pushes the assessment further. Zygoptera sit in the moderate sensitivity tier of the standard bioassessment indices, more forgiving than stoneflies or mayflies, but still intolerant of severe degradation. Their presence in this drainage ditch is, in the most measured scientific language I can muster, a pleasant surprise. The Gambusia encountered here confirm a functioning zooplankton and invertebrate food base. Something is eating, something is being eaten, the trophic structure is intact. Together, these three species describe a ditch that is impaired, stressed, but yet biologically functional. In urban ecology, that's a pretty remarkable combination. Procambarus clarkii, the red swamp crayfish. Originally a native of the American South, now comfortably colonizing every inhabited continent except Antarctica, because Procambarus clarkii does not recognize borders and has the ecological manners of a wrecking ball. It is genuinely, without irony, magnificent. Those claws are sensory organs as much as they are weapons, dense with chemoreceptors and mechanoreceptors that read chemical gradients, pressure waves from moving prey, and substrate texture. A crayfish, claws first, is reading the ditch the way you look at a book. A very complicated water chemistry book. They are aggressively territorial. That raised claw display is not a performance. It is a genuine threat assessment conducted with complete sincerity by an animal the size of about your thumb. I respect the commitment. In the aquarium, crayfish are endlessly entertaining and catastrophically hard on plant life. If you want a crayfish community tank, the answer is the dwarf crayfish. Cambarellus species. Compact, comparatively civil, and far less architecturally destructive. Not Procambarus. Procambarus stays in this ditch. Gambusia affinis, the western mosquito fish, one of the most widely distributed freshwater fish on Earth, mostly because it's being used to suppress mosquito larvae. Their resume is impressive. The ecological record, as we will discuss, is considerably more complicated than the resume suggests. That mouth is an evolutionary argument. The upturned profile is a surface-feeding specialist solution, optimized for taking food from the water-air interface, where mosquito larvae hang out, where spent insects land, where a small fast fish can extract maximum calories for a productive shallow system. They are live bearers. Females give birth to fully formed fry, not eggs. A single female stores sperm and produces multiple broods across weeks. In good conditions, a Gambusia population scales explosively. This particular trait, combined with their appetite, is why they have been introduced to many waterways worldwide and have driven multiple native fish populations to the brink. They are, in most neutral scientific language, an ecological success story with significant collateral damage. And here's our find of the day, a damselfly. Distinguishable from dragonfly, as it is much thinner, it is waiting, as they often do, to use those helicopter wings to go catch some prey. Their nymphs are also aquatic and are great ambush predators. Definitely not something you would want in your aquarium. Now for some collection, because we're going to take a closer look underneath the microscope, and we're also going to see what pops up after we let it sit for a little while. The best jars are ones that are not overloaded with organic material or debris, and have just a light spritzing of leaves, maybe some plant matter, and a little bit of that sediment from the bottom, because that's where you're going to find most of the living things. If you end up taking too much organic matter or otherwise, unfortunately your jar will not last long, as there will be lots of decomposition eating up all of that organic matter and reducing the oxygen levels in that jar to a point where they will not sustain life. Now, to test some of the water from the drainage ditch, we're using some very easily obtained and very cheap test strips. Not the best for accuracy, but it'll give us a ballpark measure of what's going on in our water chemically. As we look at the test strip, you'll notice that aside from it being quite hard and there being some nitrates and nitrites, which frankly are not to be unexpected in such a living system, there's nothing of note. No lead, no copper, none of the things that one would think would poison a water system. Now, of course, this doesn't test for everything, but I don't have a mass spectrometer or any other strange chemistry tool laying around, so you get what you get. And do note that the nickel test is looking like it's going crazy. If you take a look at the footage, it's because the water ended up mixing as per gravity from the test strip above it. One quick thing before we leave our drainage ditch: do remember to follow all laws concerning your state or national areas of waterways. There are very specific regulations on where and when you can collect anything from the water. Some of these require having a fishing license. Do make yourself aware before trouncing around in someone's backyard as well. That might not suit your neighbor. And the water goes under the microscope. The rule is, whatever's in there, we name it, and we ask what it is telling us about the water. Because at this scale, every organism is still very much a data point. Put that slide under the scope, turn it on, let's see what we got. And here we are at 40 times magnification. It almost looks like worms moving about, but there's something strangely disjointed about them. But worms they are not. They are diatoms of Bacillaria, a colonial diatom that is pretty significant in microscopy. This is the first species of diatom ever formally described as a species, and they were so baffled by its movement that they initially classified it in the genus Vibrio. And for those of you that know, that is the same genus as cholera, an understandable mistake given the era. Watch the colony. Each individual cell is sliding along its neighbors, extending the colony into a line and then contracting it back. Every cell moves simultaneously in a coordinated fashion, and the coordination has no nervous system behind it. It is chemistry all the way down, and it produces this beauty. Each cell is encased in silica, a glass box essentially, made of polymerized silicon dioxide. When Bacillaria dies, it sinks and persists in the sediment, potentially for millions of years. Diatom assemblages and sediment cores are how we read the history of water bodies going back through geological time. In this ditch, if we so took a core, it would show exactly where any sort of agricultural runoff started. The diatoms, remember. And here is where Bacillaria is telling us about the ditch specifically. In freshwater systems, it is a bioindicator of human-induced pollution. It favors nutrient-rich, eutrophic conditions and appears preferentially in waters affected by agricultural runoff, wastewater, or urban drainage. In abundance, it is not an accident. It is a diagnosis written in silicon, one cell at a time. And here we have a Paramecium, the organism that launched 1,000 high school biology units, here in its actual habitat instead of a prepared slide. In the classroom, Paramecium is a demonstration. In our water sample, it's doing its job, hunting bacteria, navigating the chemical landscape of the ditch water, performing all the behaviors we modeled in controlled conditions, but for real, in a world it actually has to survive in. The Paramecium has an oral groove, and it sweeps bacteria into the cell. It eats by making a small vortex. It is at the cellular level doing something structurally similar to what a whale filter feeding does, scaled down by several million. The principle's the same. Create flow, capture particles, and then digest them. Evolution found this solution independently at both ends of the size spectrum. The Paramecium's presence in this water sample confirms that there is enough bacterial food to support a population of filter-feeding protists, which is, in ecological terms, the bottom of the food web doing its job, decomposing organic matter into bacterial biomass, making that biomass available to higher-order organisms, and so on and so forth up the food chain. The trophic structure works because of organisms like this one, the ones you cannot see without a microscope. There we have it. 2 organisms, 2 verdicts on the same water. Bacillaria says the ditch has a nitrogen problem, a eutrophicational signature, the chemical footprint of runoff. Paramecium says there's enough bacterial productivity to support a functioning microbial food web. Between the macro and the micro, we have a complete ecological portrait of a concrete trough in a suburban landscape. I did not expect this to be the case, but I'm always delighted to be finding the opposite. After the sediments have settled for a day, we can now take a look at the other small inhabitants of our drainage ditch. What you're seeing most busy buzzying around are the Ostracods. They are tiny bivalved crustaceans, not mollusks, despite the shell. Each one is a shrimp inside of a little bean, and the bean is made of chitin and calcium carbonate, and it has been solving the problems of aquatic life in exactly this configuration since at least the Cambrian. They are older than fish. They are older than insects. They were here first, and they're still here, in this jar, in this drainage ditch, in a suburb. If you look closely, you can see their small appendages working at the shell's edge. They are simultaneously propelling themselves and trying to get food. The ostracod is characteristically efficient. It has combined locomotion and dinner into a single motion. They eat algae, bacteria, detritus, anything small enough. In a healthy aquarium, you'll find them near the bottom performing the same function. Constant, low-level, tireless processing of the organic matter that would otherwise accumulate at the bottom of your aquarium. There are also Copepods here, our friends from the previous episode, and they'll show up just about anywhere as well, even here in this drainage ditch. The Copepods' pollution tolerance varies by species, but as a group, they're pretty darn persistent. Finding them in this jar is not surprising. It is at this point simply expected. So now we have the full picture of our drainage ditch. It's impaired. It carries the chemical memory of everything from upstream. Bacillaria are telling us there's nitrogen. The crayfish is telling us the oxygen is alright. And the damselfly nymph is telling us that the water is at least functional. The Copepods, well, they'll be there no matter what. They're always there. Just remember that all of this is analogous to your aquarium. These organisms do the same function in your aquarium as they would in these streams, and the presence of some or the thriving of others will tell you lots about your water chemistry. Before we go, I found this in the water sample. It's neither Bacillaria or Paramecium. I wonder if you guys know what it is. If you do, please throw something in the comments down below. We'll see what it is on the next episode. Welcome back to Aquarium Ecology, where nature has the answers. But are you listening?